Hello, we'll wait for some time and let everybody join in, then we'll begin. Good evening, everybody. We will be starting soon. I'll wait for some more people to join in. Uh, Good evening. Welcome to the fifth session of the live interactive discussion that we have. I'll just wait for a couple of minutes just in case if somebody comes up, and then we will start. I hope you all can hear me. Anybody can respond? Okay, good. Then we'll just start in 2 minutes. Just in case if anybody wants to go join in, they can come. Okay, I think we can begin. Welcome all of you to another live interactive doubt clearing session of the course Genome Editing and Engineering in NPTEL, which is taken by the Professor Utpal Bora of IIT Guwahati. I am Sanket Mukherjee from the School of Biological Sciences in IIT Delhi. So in today's session, I will be briefly summarizing the central dogma of molecular biology about the different protein structures. And then we'll talk how these protein structures are important for their function. In this context, specifically for the DNA binding function. Then we will talk about some DNA binding, proteins like FOC1 and zinc finger domains, which has been discussed in the first lecture of this week. About the applications, I'll not discuss much today. I will have some questions, but you please read them by yourself. So talking about the first part about the central dogma of molecular biology, I think all of you know about this. It is how DNA makes copies of itself through the process called replication. So it uses an enzyme called DNA polymerase. Now this DNA forms RNA. This process is called transcription, and it happens due to the enzyme RNA polymerase. And then the RNA, it forms protein. This process is called translation, and it happens in the ribosome. Again, it's an enzyme. It's a ribonucleoprotein. It's a nucleoprotein enzyme, which works for synthesis of amino acids, which form the protein. Now, regarding the protein, as you all must be knowing that protein initially it has a primary structure, which essentially is the polypeptide chain. Then it forms secondary structure like alpha helices and beta sheets, which after forming polypeptides, they form this kind of structure. Then these secondary structure then form a 3D conformation that we call as the tertiary structure, and multiple tertiary structures they come together to form a quaternary structure, which we get as a protein. So that is how a protein is formed. Now in case of alpha helix, most of you must be knowing that it is one of the most common secondary structure. In case of proteins, there are 3.6 amino acids per turn in an alpha helix. So in let's say that is how it is. And the first residue of the first amino acid of the alpha helix, it interacts with the fourth amino acid. So it's like uh, ith residue will interact with the I plus fourth residue. It will form this, intramolecular hydrogen bonds, which will give more stability to the structure. So the NH group of the first residue will interact with the COOH of the fourth residue, something like that. So if we have something here, I don't know if it is visible in the cartoon. So see, the COOH and NH, they are, forming hydrogen bonds together. So this is how it is. Then we'll talk about beta sheets. So beta sheets, these are linear structures. If there are two or more adjacent beta sheets together, then we call them as beta pleated sheets. As you can see in the left hand side, this is a beta pleated sheet. Now, these sheets could be parallel in nature, meaning that all of them are from the N to C conformation. That could be one of the ways how they could be present. The other one is anti-parallel, where they are N to C, then again N to C. This is again in alternate fashion. They are like this. So this is anti-parallel beta sheet structure. There could be also mixed form of structure. And usually proteins, they do not only have the alpha helix or beta sheet, they form a mixture of both of these. So we'll talk about it. So this protein interaction, it drives the cellular events that happen in a cell. So DNA binding proteins, they have a very important role in genetic activities within an organism, such as if for the process of transcription, for packaging, even replication, it requires DNA binding, rearrangement and repair. So all these processes, these are very important processes. They all require DNA binding proteins, and some of these we will see these structures that the protein form. They are important for these DNA binding properties of these proteins. Let's see. So the important protein structures or functional groups which are present in a protein, which usually interact with the DNA, they are either helix turn helix or zinc coordinating motif. It could be zipper type motif. Other alpha helix, this is not A, this is alpha helices. There could be beta sheet, beta hairpin structures, other structures, and some enzymes also, which are known for interacting with. We'll see through it. So initially, in this context, the first enzyme that was found for genetic engineering was FOC1, which was isolated from Flavobacterium okeanocoides. You must have studied about it. So do you know which class of restriction endonuclease does it belong to? Anybody?
Second group, group 2.
Group 2, which subclass then?
Group 2 restriction endonucleases.
Group 2S, right? Type 2S. So this is what it belongs to. Most of the restriction enzymes that you would be using in a molecular biology lab, they belong to type 2P. So what is the difference between these two? So the type 2P restriction enzyme, they usually have a palindromic, recognition sequence, and usually they they cleave within their recognition sequence, whereas this type 2S, S stands for shifted So essentially, the recognition site and cleavage site are a little bit, farther from each other. So in this case, this is the recognition site, GGATG of FOC1, and it cleaves 9 to 13 bases away from its recognition site. So that's the idea. We'll see how this enzyme works. Now, regarding FOC1, its molecular mass is 65.4 kilodalton. It has 587 amino acids. And it's, as I said, FOC1 is a type 2S restriction endonuclease, and it consists of two distinct domains. One is the N-terminal domain, which is the DNA recognition domain. It recognizes which, the DNA where it needs to bind. And there is another domain, that is the C-terminal domain, which needs to cleave the site. I mean, it will cleave at the restriction site. So these two domains are important. Why are we studying about this? Because later in this course also, even in some lectures, you must have heard that we create chimeric enzymes. So we, let's say, take the recognition domain from one restriction enzyme, and we take the cleavage domain from another restriction enzyme, and we get targeted genetic modifications we can get through this method. That's why it is important to recognize which domain has which function. So cleavage only happens upon dimerization, when the recognition domain is bound to its cognate site and in the presence of magnesium ion. So in most of the restriction enzymes, I mean almost all, they require magnesium ions for functioning. So uh, because of this modular nature, that there is a separate, restriction domain, there is a separate recognition domain. Because of this region, we can develop enzymes. This has given us the ability to develop enzymes based on our own specificity. So let's say if I want enzyme which cuts 9 to 13 base pair away from some other recognition site apart from what we have in FOC1. So we can create a chimeric enzyme where we will use another recognition domain of some other restriction enzyme. That's how we this kind of things happen. So here you can see that the two cleavage domain, they need to dimerize to cleave the DNA. If this dimerization does not happen, there will be no cleavage. So that's the idea. Now we will talk about zinc finger proteins. Only these two proteins have been discussed. I mean, zinc finger proteins and the FOC1, these two have been discussed in this week. So uh, they are approximately 3% of human proteome and are most common DNA binding motif in eukaryotes. And initially discovering them was also difficult because they had these zinc, involved. I mean, zinc was there. So whenever any reducing agent or, chelators were used, uh, the conformation of these enzymes, they used to change. So that's why it was discovered later, but it is one of the most common DNA binding motifs in eukaryotes. So transcription factors, they often use zinc fingers to mediate protein DNA interactions. We'll see how zinc fingers look like. So before that, among zinc fingers, there are multiple kinds, like C2H2 family is one of them, then gag knuckle, trap clef finger, zinc ribbon, and similarly, there are many families of zinc finger proteins. C2H2 is the one of the most studied ones and the most found ones also in eukaryotes. So initially this was identified through transcription studies in an African clawed frog called Xenopus laevis. Please remember this, in the laboratory of our Aaron Clark. So a typical C2H2 zinc finger contains a 28 to 30 amino acid sequence, including two conserved cysteines and two conserved histidines. These are important because these will be the residues that will interact with the zinc finger moiety. I mean the zinc ion. But other combinations are also possible. They are also present. So the regulation of transcription is seems to be one of the most important tasks which is performed by these zinc finger C2H2 zinc fingers. And since transcription is something that happens in all the cells, so their abundance is also pretty high. So let's see how they look like. So if you see, as we already said, that they contain 28 to 30 amino acids, right? And their characteristic structure, it includes a beta hairpin. As you can see, this is the hairpin moiety that has two anti-parallel beta sheets. So we have already studied how they look like. Anti-parallel means one will be like this, another one will be like this. So one will be, if this is N2C, another one will be N2C, like this. So, and in between these beta sheet, there will be an alpha helix. So that's how the structure is. So this is the alpha helix, then there is the beta sheet. And as we already said that the two cysteine residues, as you can see here, and two histidine residues, they are conserved. Why are they conserved? Because they are important to interact with the zinc, which is present in between this hairpin loop. So what will happen if I use a reducing agent, or let's say if I use a metal chelator which will remove this zinc out of this enzyme, what would happen? Will it retain its activity?
No, sir.
Yeah, correct. So this is very important that you understand this. So the zinc is there for for the stability of this protein, and to maintain its function, this zinc is very important. So that being said, so zinc, it does not interact directly with the DNA. Alpha helices actually do in this case. They insert into the DNA major groove where side chains form specific contacts with three consecutive base pairs. The overall interaction is further stabilized by some non-specific contacts between the beta sheet and the sugar phosphate backbone of DNA. We have one question regarding this. We'll see how it works. So regarding zinc finger modules and binding site, a canonical zinc finger module consists of two antiparallel beta strands and an alpha helix with zinc iron, which is coordinated by conserved cysteine and histidine residue pairs. We have already discussed about it. Various amino acid residues at positions relative to the amino terminal end of alpha helix form important base specific contacts in the major groove of the double stranded DNA. So essentially, what amino acid residue you have in the alpha helix, that would dictate the recognition site of this protein. So that is important. So initially, these zinc fingered proteins, they have non-specific binding, then they have some semi-specific binding, and then they go for specific binding. We'll again see how that happens. So let's answer a few questions. And since this is an interactive session, I would expect that you all give some answers to these. So let's see if you can do it. I'll maybe give you a couple of minutes for each question. So the first question is, which is the most common type of secondary structural elements in proteins? So is it alpha helix, beta sheet, parallel beta sheet, or anti-parallel beta sheet?
Alpha helix.
Alpha helix. I hope it is clear to everybody. We just discussed a while ago that alpha helix is the common type of secondary structural element in proteins. Then we will talk about zinc finger were first identified in a study of transcription in the species of African dwarf frog, African clawed frog, African threadfish, or African wild dog.
B.
African clawed frog. Can anybody frog? Can anybody tell me what is the exact name of this species?
Xenophus leaves.
You are absolutely right. Uh, Next, let's go to the next question. So the proposed sequential binding scheme of the three cysteine, histidine, zinc finger motif, uh, domain is here. Bound to the cognate DNA are specific binding, semi-specific binding, non-specific binding, or non-specific binding, specific binding, and specific, then semi-specific binding, or non-specific binding, semi-specific binding, and then specific binding, or binding, specific binding, semi-specific binding. Which one will it be?
C.
You are absolutely right. We have discussed about these things. So these are some conceptual questions. So this is how it looks like. So initially there is non-specific binding. So because of the charge of these proteins, they interact with the DNA in a non-specific manner. Then there is some specific binding that happens, followed by which they are bound to their cognate DNA. Cognate DNA in this case means the recognition element where it is supposed to bind. So how will the specificity of this recognition site be determined in a zinc finger?
Can you repeat the question?
How will this will this zinc finger bind to? How will that specificity be determined?
The sequence is present in the target gene.
Yeah, that is correct. So it will bind to some sequences in the target gene. So you have multiple zinc finger domains. Which particular feature of this zinc finger domains will make it specific to a particular target gene and not some other gene? That's what I'm asking. So what does a zinc finger protein contains? Alpha helix residues, and there are two beta sheets which forms the hairpin structure. Correct? Am I right? So which one of these will be involved for the specificity?
Alpha helix.
That is correct. That is the alpha helix residues. They will be involved for the specificity of this zinc finger domain. So yeah, you're right. So which is not the current technique used for gene delivery in zinc finger nucleases? Viral vectors containing adeno-associated virus vectors, AAVs, or adeno virus vectors, ADVs, and lentivirus vectors, LVs, etc., or non-viral vectors which contain polymers and cell-penetrating peptides, etc. Physical methods containing microinjections and electroporation, or GFP or molecular scissors. Which of the technique it cannot be used for gene delivery? That's the question.
TGFP or molecular scissors.
Okay, so what are molecular scissors?
Restriction enzymes.
You are right. So these are restriction enzymes or GFP. So the current techniques which are used are viral vectors. So these are viruses which are modified, where we disarm these viruses for their virulence, and we then transfer a gene. So very similar kind of things we like adenovirus vectors have been also used for COVID vaccines. So that is how you deliver genes, and these genes are then transcribed and translated into the host wherever you are, delivering them. There could be non-viral vectors, so they could be polymers like polyethylene amine or polyl-lysine, or liposomes also, cell-penetrating peptides. These kind of things can be used. Some physical methods like microinjections, electroporations. Nowadays, even among the physical methods, sometimes even nanoparticles are used. So these are the different ways how you can deliver your gene of interest into the cell. So that is how what it I mean this is all about. And GFP or the restriction enzymes, these are not involved in gene delivery. Rather, these are involved for gene modification or tagging, like GFP is used for tagging the genes, so something like that. So the CCR5 del 32 mutation was initially discovered in 1996 as a genetic mutation that confers protection to cells from infection of coronavirus, HPV, HIV, or smallpox. What is HPV?
Human papillomavirus.
That is correct. That is correct. HIV is human. What is HIV? Human immunodeficiency virus. Yes, yes, correct. Very good. So which of these viruses, which of these viruses cannot, this mutation will confer you protection against this virus. Which of these virus?
HIV.
HIV. So exactly. So CCR5 is a co-receptor of this virus. So if CCR5 is modified, then the viral replication is affected. We'll see how that happens. So virus, it binds to the glycoprotein receptors. If you can see here in the figure, the A section, it binds to the glycoprotein receptors of the host cell CD4 and co-receptors CCR5. So which cells have CD4 receptors? Anybody who knows something about immunology?
T4 cells.
T4 cells.
T helper cells. Sorry, T helper cells.
T helper cells, right? TH cells. So TH cells they are important for they have an immune function. So this virus, it binds to one of the receptors, which is importantly mainly present in the immune cells, that is the TH cells. It binds to the CD4 receptors, and the co-receptor is CCR5. So it has been, so what happens? They have GP41 and GP120. These are glycoproteins present on the surface of the virus. They help, them to attach to these CD4 and CCR5. Now, when it gets attached, then the host cell membrane, it helps in internalization of the virus. It gets inside the cytoplasm. Once it gets inside the cytoplasm, what happens? The virus, it disintegrates, and then the viral whatever DNA is present, in this case it would be RNA. So viral RNA, it would be converted to DNA, and then it will transcribe itself, and it will form the proteins which are essential for the virus to multiply. So essentially more number of proteins will be formed, and these viral RNA and proteins, they will assemble together, and then they will go out of the cell to infect more cells. So they will infect more TH cells, more immune cells. So slowly your immune system will be affected. These immune systems will no longer function, because when these viruses, they multiply, they hijack the immune cells machinery, their function will be, their efficiency will be lowered. Hence you will no longer have the immunity that you used to have. So in that case, that is why these are called human immunodeficiency viruses. So this virus, it causes severe immunodeficiency in the patients where it infects. So it has been observed that the CCR5, which is a co-receptor, it was not known earlier. So this co-receptor is very important for this virus entry. So if the virus does not enter, if this co-receptor is modified, if the 32 base pairs, these are deleted from this co-receptors, then it is modified and the virus can no longer recognize, it can recognize the CD4 receptors, but the internalization of the virus is no longer happening. That's why they do not replicate, and it provides a protection against this virus. So that's the whole idea. Does everybody understand how this is working? Okay, good enough. Even there were some, trials. I don't know whether you know this, that CCR5 mutants, they tried to create, in China, but there were some ethical issues that came up. So, I don't know whether you know this, but just for the sake of knowledge, I'm telling you all. So in China, they tried to create this mutant. But again, if you do something like this in humans, so to test whether your mutant is working properly or not, you need to infect the person or the patient in this case with HIV, which is again very unethical. So
Sir,
which
I
of the
just have.
fall? Yes, yes.
Sir, if we do the CCR5 into mutant, what about the original functionality of the particular co-receptor? Will it not affect other
See,
viruses? Recognition?
other viruses are not known to see different viruses. They have different mode of internalization. So like spike proteins, they, in case of COVID, SARS-CoV-2, it binds to certain receptors which are present in the, your respiratory tract. So even influenza virus, they have like a sialidase enzyme. So we have seen when we study about each and every virus, if you study how it is, that is why it is important to know how these viruses function. So if you knock out this sialidase enzyme, then what will happen, this virus will no longer be able to infect you. In case of influenza virus, I'm telling you. So there are similarly there are studies which are different for different viruses. So HIV infects CD4 and CCR5, the receptor and co-receptor. Not all viruses do that. So, but you're right, probably if we mutate it, maybe it will be beneficial for some other virus. We don't know that yet. So, again, so that's why it's unethical in a lot of aspect. So there are many problems that could come up. Does that answer your question, or was it something else?
Yes, sir.
Okay.
Yeah. That is my question.
So which of the following is famously known as the guardian of the genome? Is it P63, D53, E6, or P53?
P53.
You're right, it's P53. So P53 is a tumor suppressor gene. I think a lot of you know about this. So in a lot of cancers, this gene is, it's knocked out or knocked down, rather, in cancer cell lines specifically. So there are mutations to this P53 genes, so that make those cell lines oncogenic. So something like that. So it's a 53 kilodalton tumor suppressor protein, which decides the fate of the cell during stress conditions, and that's why it's called as the guardian of the genome. So that's the thing. Zinc finger nucleases, they were first used against DASH, which prevented the virus from replicating within the host cell. So was it HPV 1618, HPV E2 gene, HPV 16E6 gene, or HPV P63? Does anybody know? Okay. I'll give maybe 1 more minute for you to think. If anybody needs to give any answers.
D.
I'm really sorry. B. Okay. I think there was a network issue. Am I audible? Hello? Yeah, I think I'm audible. Sorry, it shows a bad network connection here, so that's the reason. You are right. HPV-E2 gene was the one where ZFNs were first used against, which prevented the virus from replicating within the host cell. It was discovered by Mino and group in 2009 initially. So this is how the genome of HPV it looks like. It has E1, E2, E4, E5, all these genes. So all the E1 to 7 genes, these are the open reading frames, which are the early region of this virus. And the late region, it has the L1 and L2. So the difference is that these are, the E genes, they will be expressed first, and then the late genes. This is how it is, basically how it works. So E2, the early genes, a lot of them are on HPV, they were carcinogenic. So that's why, uh, you know, uh, devising a strategy against them uh, helps in preventing the virus to cause cancer. So nowadays, even E6 is used as a target. You must have read about it in the lectures. So ZFN was used for that. So that's the question. Now, question 8. Which of the following is incorrect in ZFN mediated targeted genome modification while double-stranded break repair? A pair of zinc finger nucleases binds to a target sequence, dimerize through the FOC1 domain and join the DNA at defined site, or the double-stranded break generated by zinc finger nuclease zinc finger nuclease cleavage induces the DNA repair process. In the absence of donor template DNA, error-prone non-homologous end joining result in targeted mutagenesis, or in the presence of homologous sequences, homology-directed repair result in gene editing or site-specific integration. Which one is incorrect in the following four statements?
Yeah.
You are right. You are right. So what happens? What would be the correct one here? A pair of zinc finger nucleases. They do, yes.
Cocoa domain produces double strand break on join.
They produces doubles. They cleave essentially. Yes, you're right. So this is how zinc finger nuclease mediated targeted genome modification looks like. So a pair of zinc finger nucleases, they will bind to the target sequence. They will dimerize through the FOC1 domains and cleave the DNA at the defined site. So which in this setup, which is the recognition domain and which one is the cleavage domain? Can anybody tell? Anybody?
Fork one is the cleavage domain, and what are all the fingers are recognition, they are definition.
So this is the concept of chimeric, restriction enzymes. So we create this kind of thing so that we can have more, control over targeted gene editing. So that's the idea. So let's complete this, then we will talk a little bit more. So the double stranded break, now once generated because of that cleavage, uh, induces the DNA repair process. Now you know how DNA repair processes happen. So if there is a homologous strand, then there will be homologous directed, repair. And if there is no homologous strand, then there will be non-homologous end joining. You have already studied about it in the DNA repair part earlier. So that would be all for today. So just as a question, I would like to ask you. So uh, these were the ways which how people used to edit genes specifically using creation of chimeric enzymes. So anything the one of the most modern ways how people do it now? Just a question.
I think is it CRISPR Cas9?
Exactly. It's CRISPR-Cas9. Actually, not only Cas9, there are many other proteins which are involved. Cas12 has been discovered, Cas14, so many proteins. So Cas is important for the cleavage activity of this enzyme, and CRISPR is for the recognition activity. And the beauty of this entire system is that you can, you just need to create a guide RNA that will to creation of guide RNA will help you to recognize any particular sequence uh, in your genome, and of course the Cas proteins, they will help you in cleaving. So it has made this entire process of genetic gene editing much simpler. So we'll be I mean you will be studying about it in the later weeks, but that's all I have for today. I'll just repeat the questions once more, just in case if anybody has missed any point. So the first question was, which is the most common type of secondary structural element which is present in the protein, that is alpha helix. And then the zinc finger, they were first identified in a study of transcription in the species of African clawed frog, specifically Xenopus laevis. And then we had, the sequential binding scheme is first there is non-specific binding, then semi-specific binding, and then specific binding for zinc finger domains. Then this is the schematics, and which is not the current technique for gene delivery. We discussed GFP or molecular scissors. They are not the current technique. So the CCR532 base pair mutation, which was discovered in 1996, it confers protection to the cell from HIV infection. So we have already discussed how that happens, how HIV it replicates in the cell. So we discussed about it. Then we talked about the protein which is famously called the guardian of the genome, that is P53. So that we have discussed. So we also said that zinc finger nucleases, they were first used against the HPV-E2 gene. HPV is human papillomavirus. E2 is an element of this gene, of this virus, uh, which is an early, ORF or transcript. So against this, it was a device. Zinc finger nucleases are devised, and it was observed that the replication of this viruses were affected. Then we talked about zinc finger mediated targeted genome modification. So we talked about it in detail in the schematic, how it works. So that would be all for today. Do you have any more questions? I'm ready to answer them.
Sir, in the zinc, the ZFN mediated targeted genome, that we are introducing, say that sequences now for homologous recombination. Those are exposed to exonucleases in the cell. How it is getting prevented by exonucleases activity?
Really not
We are
understanding
giving,
it.
yeah, we are giving donor sequences now. So for homologous recombination, the genes which we want to knock in or knock out. That one, how it is getting prevented from exonucleases activity inside the cell.
So usually, as far as I know about the experiments, how we perform these experiments, sometimes they are done in vitro. So in vitro systems, there is no problem of introduction of exonucleases as such. So since if we do not give such enzymes, there will not be any problem. So I
So,
think
but still in cultured cells, they'll have some exonucleases inside. So, what
so
about the activity of
in
how
what
they get the
I see, the thing is, the problem with any kind of, gene editing technology in a cell culture system is, I'll tell you why it is problematic. Because not because of endonuclease or exonucleases in specific, but because let's say if you are doing something in vivo for a mouse, you will you will have to do these modifications in the embryonic stage. We have talked about it, I think, previously also. So there you have to find which of the cells they actually really transform. Where you'll transfect these cells, you'll edit them, and you'll edit the gene, and you'll introduce the desired mutations and stuff, and then you will transfect them, and you will see which of these cells have actually transformed, and those cells you'll have to then plant it on a foster mother into the embryo, and then in a hope that all the cells will have this kind of a modification. But if you use these kind of techniques for curing a disease as such just in a human or even in mice, so you are only targeting one cell. You cannot do this for multiple cells altogether. Anyway, these have very low efficiency, so that's the problem. So are you understanding?
Yes.
So the efficiency factor definitely comes due to a lot of things. Whether if endonuclease is a part of it or not, I'm not sure. I'll have to look into it. But see, the things are not as simple as you just want to create this kind of a change, and it just happens right away like that. Such things really don't happen. The efficiency is usually low. So, but in vitro systems,
So
this is
sir,
good.
even in successful attempts, is it due to the luck, or we are doing something to prevent that enzymatic activity? That is my question.
As I told you, we edit the gene first, and then we transfer this, edited gene into the cell. That is how the process works. Are you understanding?
Yes, sir.
So that is how it works. Sometimes on even on a cellular level, sometimes even if you have like a few cells which are transformed, which have your edited gene, if they are replicating more in number, so they will form the major population. You're understanding something like this. You need to devise a way how your desired mutation it exists in the population. So these are some problems which happen in routine cell culture experiments.
Thank you, sir.
Anything else? Any other question? That is a very good question. I really like the question.
Thank you, sir.
Anything? Any other question? Okay, so I hope the contents are clear to all of you then. Please at least react or something. I'll understand that you're listening. Okay, okay, thank you. So I think if everybody is okay, then we can end this session here, and you people can leave. We will call it a day. We'll be meeting next Tuesday, same time. Okay, thank you. Thank you very much for joining in. Hello, Raj. Do you have any questions? You just joined in. Am I audible? Are you here, Raj? Should we? Okay, for your sake, I will just repeat the questions that we had discussed today, since you have joined. Maybe we'll start from the beginning. Maybe. So we talked about that central dogma first, how DNA, it forms DNA, and then it forms RNA and protein using enzymes like polymerases and ribosome. In this case, I hope you know about it. If you have any questions, you can raise your hand. Then we talked about protein structure. Then about how primary structure, it is usually a one-dimensional structure. Then it forms a 2D structure, secondary structure, which is formed of alpha helices and beta sheet. Then there is a tertiary structure, which is a 3D structure, and about the quaternary structure. So alpha helix is one of the most common 2D structure. Then there is beta sheet, where it could be a parallel sheet or an anti-parallel sheet. So we talked about different DNA binding domains. So we talked about fork1, how it binds to DNA, its recognition site. We talked about them, and how it cleaves 9 to 13 base pairs away from the recognition site. So and then we talked how these cleavage domains, they need to form a dimer to cleave, in case of fork1. Then we talked about zinc finger proteins, which were isolated first from Xenopus laevis, and they contain, conserved cysteine and histidine uh, residues that interact with the zinc in the hairpin loop. Now, this hairpin loop, it consists of beta sheets. One is parallel, another one is anti-parallel. So we talked about it. So then we talked about the zinc finger proteins, and uh, I mean, that's all. Then we discussed few questions on this. So I think it's clear to everybody. If it is, you can leave. That's all for the session. If there are any doubts, you may ask.